Right. So, welcome everyone to the PSI Virtual Talks. It's co-sponsored by the Center for Translational Research in Neuroimaging Data Science, the Trend Center, and the IEEE Signal Processing Society. I'm Roger Silva, and a research scientist here at Trends, and today it is my privilege to introduce our speaker, Dr. Mandy Mejia. She is an assistant professor in the Department of Statistics at Indiana University. Her research focuses on the development of statistical techniques for the analysis of fMRI data, with the goal of extracting accurate insights into brain function and organization at the subject level. She pursues that with her group by developing computationally efficient Bayesian techniques and statistically principled noise reduction approaches, performing these analyses on the cortical surface and subcortical gray matter to facilitate spatial modeling and intersubject alignment. Her group also produces open source software, including the CIPSI tools R package for handling and visualizing cortical surface data in R. Dr. Mejia has applied her work to amyotrophic lateral sclerosis, thalamic organization and connectivity, autism spectrum disorder, and Alzheimer's disease. Mandy is an accomplished, very active researcher pushing the envelope in fMRI research with new and improved methods. Today, she will be presenting on template-independent component analysis using empirical population priors to extract more accurate subject-level IC maps from fMRI data. Dr. Mejia, it is an honor to have you here today. Thank you so much for sharing your time with us. You have the floor.
Thank you. It's my pleasure to be here. Can you hear me?
Yes.
Okay, great. Thank you, Rogers, for that very nice introduction and for the invitation. I'm really happy to be here sharing this work with you all. It's going to be, I think it'll be pretty informal. I will be giving a very kind of high-level introduction to this approach, and then actually sharing some of our ongoing reliability studies, as well as some of our published reliability studies as well, and then discussing some extensions that we're currently working on. So to give you an overview, I will give an introduction to this, this framework that we call Template ICA. And then I'll follow that with a very brief description of the R package, the Template ICA R package, where you can apply Template ICA, hopefully in a user-friendly way. And then we can take a quick break there, or a long break. I'll let Rogers decide. And then I'll go into a couple of reliability studies that we've performed to evaluate how Template ICA actually performs in practice. I should say we have lots of simulation studies as well, but I'm not going to show those in this talk, but they are in our original paper. So if you're curious about simulations, I would refer you to our paper, or I'm happy to talk about it later. And then I planned for 2 Q&A breaks during those reliability studies, one after the first reliability study and the second after the second reliability study. And then I'll just briefly describe some extensions and ongoing work that we're currently pursuing. Okay, so to give you an introduction, so I am sort of assuming some familiarity with ICA, but here's just a kind of brief background slide. So the goal of ICA in general is, when applied to fMRI data, is to decompose the data Y, which is a T by V matrix, into a matrix of independent components. So here we have L independent components, and their mixing matrix, which contains the temporal activation profiles of each independent component. And we assume that the rows of S, each row of S, represents either a brain network, some kind of functional area of the brain that's acting in a coordinated manner, or some source of noise in the data. And so this, you can simply, very easily run an ICA estimation on fMRI data, but single subject ICA applied kind of out of the box has a number of important limitations. First is that the data has very low signal to noise ratio. So we tend to get very noisy ICs that tend to often represent a mixture of signal and noise components. And then it's difficult, if not impossible, to match those independent components correctly across subjects or studies. And so an alternative that many people use kind of in a general way is to start with a group level ICA, where we identify group average independent components, and then to obtain subject level independent components kind of post hoc. So we are going to fit into that family of alternatives, but there's a number of methods that fit within that kind of basic approach. So I could categorize those types of methods into roughly kind of 3 groups. The first I would call ad hoc approaches. For example, dual regression, where we simply regress the group level ICs against the subject level fMRI data to get the subject level mixing matrix, and then regress the mixing matrix against the fMRI data to get the subject level ICs. And this is a very popular approach. It's very computationally convenient, but it lacks much justification. So it's an ad hoc approach. The second is what I would call optimization approaches. For example, most notably, I would say group information guided ICA. And the goal of group information guided ICA is to perform a single subject ICA, but while introducing a penalty parameter describing the distance between the subject ICs and the group ICs, so that the subject ICs are matched to those existing group ICs. And perhaps you get a bit of kind of shrinkage towards the group ICs as well. And then the final category I would call statistical approaches. So for example, template ICA, which I will be describing in this talk. Okay, so here's a kind of more detailed comparison of these 3 approaches that I've just mentioned. So we have dual regression, group information guided ICA, and template ICA. So again, the basis of the approach for dual regression is that it's based on these 2 regression models. Group information guided ICA is an actual ICA model, kind of an almost traditional ICA model, but with a penalty for the distance from the subject to the group ICs. And template ICA is a statistical model that performs shrinkage of subject level ICs towards group ICs. And these 3 approaches have some things in common, but some important differences. So the first thing they have in common is that the subject level ICs are matched to the group ICs and therefore across other subjects and other studies that are estimated using the same approach. All of these approaches are also applicable to new subjects. So the subjects don't have to be included in the group ICA estimation for these methods to be applied. And so that makes them very convenient. In terms of noise reduction, dual regression does not do any real noise reduction. Group information guided ICA may do some noise reduction depending on the strength of the penalty, penalizing the distance to these kind of nice clean group ICs. Template ICA does do noise reduction, and this is determined by the template variance, and we'll get deep into those weeds later in the talk. The use of a template, and what's the definition of a template? So dual regression does not, I guess you could say they use a template as well, the group ICs, but they don't really think of it that way. Group information guided ICA uses the group ICs as the template as kind of the target to shrink towards. Template ICA estimates the mean and the between subject variance of those group ICs, and that is what constitutes the template. So it's more of a statistical prior. And then 2 features of template ICA that are kind of unique to our approach is first, template ICA has a built-in procedure to remove nuisance ICs. So if the data has not been kind of scrubbed and fixed and all of that, there is a step for removal of nuisance ICs built into template ICA. And so that can help with noise levels as well. And then finally, because template ICA is a statistical framework, we can perform inference to do things like principal thresholding of the subject level IC maps. So again, we'll get into all these weeds throughout the talk. Okay, so the basic template ICA model is simply a single subject ICA model with this additional level. So we have on the first row is really just a traditional probabilistic ICA model because we have a residual here at the single subject level. And then the second level is, so it's kind of a hierarchical model, is how we believe those subject level ICs relate to the known group ICs. Pardon, I have to not touch my mouse when I'm using Google Slides. So we're assuming here that the subject ICs can be modeled as a deviation from the group ICs. So we have this deviation term delta, and these group ICs S naught. And the deviations delta are assumed to be normally distributed with mean 0 across subjects and a known variance that is going to vary spatially as well as across ICs. And this variance, as well as the mean and SO, that is again what constitutes what we're calling the template here. And so in this talk, I'm not going to give a lot of detail about how we estimate this model, but it's actually pretty straightforward and computationally convenient. Basically, we use an expectation maximization approach. And all those details can be found in the paper I'm citing here at the bottom. And again, I'm happy to give more information about that in the Q&A or after the talk. So one thing I haven't mentioned here is that the model actually is not quite as simple as I would have drawn here. We also assume that there are nuisance components that could be additional sources of neuronal variation that are not included in the template, or noise components. So for example, I may have a template that only includes a few components that I really care about estimating. And I could say everything else that might be going on in that subject's brain, I'm going to consider a nuisance component. But even if we included all of our kind of group ICs, all of the estimated resting state networks that we know about in the template, we would still potentially want to allow for some nuisance components to absorb different sources of noise in the data. So we know that ICA is a very effective method for identifying noise signals in the data. And so these nuisance components serve an important purpose here for absorbing that noise. And so again, I'm not going to go into detail here about how we deal with those nuisance components, but I have an appendix slide, and I'm happy to talk more about that if someone's interested. Okay, so I've told you that we have this template and we assume that it is a known quantity, but we actually have to estimate it in practice. So how do we do that? So there's kind of two ingredients that we need for template estimation. The first is a set of group ICA maps, which I'm showing here on the left. And the second is a training data set of fMRI data. And so that data could come from some kind of open repository if the data is relatively comparable to the data that you're using that you want to analyze. So you could use the Human Connectome project if you're using an HCP-like protocol. You could use the Abide if you're studying a population with involving people with autism, the Baby Connectome project. So there's all these repositories that we could potentially draw from in order to establish templates. But often we want, maybe we have a population that it doesn't have a repository. For example, people I work with in ALS, there is no ALS repository that I'm aware of. So it's also possible to use a holdout data set for the training data. And so by combining those group ICs with the training data, we get a set of noisy subject level IC estimates. And it's important that we have two of those for each subject. And we say test retest here, and that could be that we truly have test retest data, but it could also mean that we take a single session of data and we split it down the middle and call that kind of pseudo test retest data. And so if we have a set of repeated measures for a number of subjects, we can mathematically estimate the mean across subjects as well as the between subject variance. And that variance that I mentioned, the between subject variance, it's important to distinguish that between subject variance from the kind of overall variance in the estimates, which also includes noise variance. So in our estimates, we have some true amount of between subject variation, and then we also have noise. And so here we're trying to isolate that between subject variation. So this is how much do subjects vary in their intensity in a particular area of the brain for a particular IC after accounting for the variation that can be explained by the noise. And so here what you see is this pattern of high variation in exactly the areas of the brain that tend to be engaged on average across subjects. So in kind of background areas that are dark on the mean, we see that those same areas have very low variance across subjects. And you see that pattern persist across independent components. And so these kind of spatially varying prior variances are going to allow the data to speak more in these areas where we know there are true differences across subjects. And it's going to perform more kind of noise reduction and shrinkage in these background areas where we know that there's not a lot of real variation across subjects. Okay, so when you run template ICA, you get a number of things out. You don't just get a set of subject level IC estimates out. So for an individual subject, when you apply template ICA, you'll get a set of estimated independent components. So going back to the model, that's this S matrix. You'll also get a set of standard errors of those estimated ICs. So these estimated ICs, both of these first two terms are based on the posterior distribution of this S, the rows of S, because this is a Bayesian model. So we have a prior, we have a likelihood that comes from our data, and so we get a posterior for the quantities of interest. And so these estimated ICs are based on the posterior mean, and the standard errors of those ICs are based on the posterior variance. In addition, we get something else that's kind of interesting. If you go back to the model, you'll see we have this S matrix that we want to estimate, but that S matrix is determined by estimating this delta matrix because S naught is known. So if we know S naught and we know delta, then we know S. And delta represents the differences between the subject and the population average. And so that could be like more intensity of engagement or less intensity of engagement, for example. And so when you run template ICA, you'll also get those estimated deviations, which we often call subject effects. And again, that's based on the posterior mean. And then you can also get standard errors of those deviations. And again, that's based on the posterior variance. And because we get both estimates and standard errors, we can perform inference. And inference can serve basically two purposes. The first is to quantify the size of engagement or the size of deviation. And the second is to perform principal thresholding of these maps. So for example, I might choose, I might want to threshold an individual IC map for a single subject, or I could construct a union mask of considering all the vertices that are engaged for any subject to get kind of a group level principled mask. And so to perform inference, we would simply use our estimates and our standard errors of either the ICs or the deviations to perform a t-test at every vertex, and then do some kind of sensible multiple comparisons correction, such as FDR correction. Okay, so that concludes the overview of the template ICA approach. I just want to describe its implementation very briefly in this R package that we call template ICA R. So there's three kind of main steps in the process of applying template ICA. The first, and I'm actually skipping the 0th step, which is to estimate the group ICs. So assuming you have a set of group independent components, you have your training data. I mentioned these two ingredients previously. The first step is to estimate the template. And so we have a function to do that. We also have a wrapper function for SIFT format data and a wrapper function for NIFTY format data. Once you have that template estimated, that can be passed onto the main function, template ICA, along with a fMRI data from a new subject that was not included in the training data. And so that is going to perform all the model fitting. And again, we have a wrapper for SIFT data and a wrapper for NIFTY data, but you can also just use the basic function if you have your data already kind of in matrix format. And then the last step, kind of an optional step, if this is something useful or of interest, is to identify these areas of engagement through the activations function, and again with these wrappers for SIFT and NIFTY format data that will actually format the output into a ZIFT object or a NIFTY type object. So I mentioned a lot of stuff with SIFT file types as well as SIFT objects. So Template ICA interfaces with this other package that we've developed called the SIFT tools package. We do a lot of work with surface format data and greater than inch format data, and so we needed something to make our lives a little easier to work with that data and visualize it. So the SIFT tools package allows for reading, writing, analyzing, and visualizing SIFT format data in R. And so, apologies. And so we could use the view ZIFT function to visualize these results directly within R, or we could also invoke the write SIFT function to write out a SIFT object so that we can later go and open up Workbench or whatever our preferred software is for visualization. Okay, so I am going to stop here for the first Q&A break.
All right, thank you. Yeah, so we have here a question already from Thomas. Thomas is asking, does this require Neuroconductor, or is it all independent?
Yes, that's a good question. Neuroconductor is a pet project of a good friend of mine. So no, it doesn't require Neuroconductor. We're in the process of trying to get Template ICA onto both CRAN and Neuroconductor. Currently it's available through GitHub, but we're actively working on that. And Neuroconductor is, I mean, it's a couple of things, but it was kind of envisioned as a repository. And so it's not like usually a requirement, but there is a kind of, I think the way that they set this up is kind of like the Tidyverse package, if you're familiar with that. Like Tidyverse, in that if you install Tidyverse, you get all these packages installed for you. If you install like Neuroconductor, you get all these things installed for you. But no, Template ICA is standalone. We do, I think, will require SIFT tools to be installed if you're using the functions that depend on it, and any other dependencies are automatically installed.
Great, thank you. So a follow-up question, well, not a follow-up, but another question that's from me. What is the relation between the posterior mean and the S0 in the, I think it's in the model of on the second level model?
Yeah. So I can answer this from a general Bayesian perspective. In our model, it's not, there's no like closed form solution, but I can give a basic intuition. So in a Bayesian model, we have basically three distributions. We have the prior distribution on the unknown parameters, and so that prior in our case is centered at S0 and has variance given by these values. And so we have the prior distribution, and then we have a likelihood, and the likelihood describes the likelihood of the parameter given the data. And so in a frequentist perspective, when we don't have a prior, we would just base our estimation on the likelihood. So in a Bayesian context, we instead have this third distribution, which is the posterior, and is typically somewhere in between the prior and the likelihood. And so it combines information from both. And how close it is to one or the other depends upon the strength of the prior and the strength of the data. So the more data we have, the stronger the likelihood and the less influence the prior has. The less data we have, the stronger the prior relative to the data, and so the more power the prior has. And so typically this posterior mean is going to be something like a linear combination between the likelihood-based estimate of S and the prior mean. But in this model, we don't have a nice closed form solution.
Mm-hmm. Okay, yeah, so that's what I was getting confused. the S0 is your prior, so it's not being updated.
Right.
Okay. Yeah.
Yes, S0 does not change. We estimate it before fitting the model.
Thank you.
Yes, well. So I guess we could say that if we had data from multiple subjects, if we wanted to fit data from multiple subjects, we could estimate S0 through the model. So that would be like more of a traditional hierarchical ICA. And my co-author here, Ying Guo, has done a lot of work in that area. The problems with taking a hierarchical approach are that, first of all, computationally, it's very expensive. Secondly, because you don't have these known group ICs, you have to model them. So the way her group has approached it is to use mixtures of Gaussians. And if you use mixtures of Gaussians, the computation kind of explodes. And so typically you have to limit yourself to 2 components in each mixture, and that's not very flexible. And then thirdly, you're kind of limited in terms of, so I guess there's 2 more points. Third is that you're limited in your size. So you can't have too many subjects, but you also can't have too few subjects, because if you have too many subjects, it's not computationally tractable. If you have too few subjects, your prior, your group kind of quantities aren't going to be estimated very well. And then the final problem is that we still would have the issue of not being able to match across studies, right? So every hierarchical ICA model is kind of a world by itself, and it's going to be hard to compare.
So here, basically, like I said, this is a subject level ICA, and it's hierarchical within that subject itself. It's not, except for the prior, which you estimated from another prior data set.
Yes.
It doesn't combine data from other subjects at this level, at the subject level.
Right. So the kind of all the data, all the combining across subjects is done at the empirical prior estimation stage.
Yeah. Now I see the parallel more clearly with Giga ICA that you mentioned in the beginning. That one is also
there's a lot of similarities.
Subject level ICA, and it's not combining except for the reference.
Yes.
Thank you. I have here another question, and that's a very traditional question. That's from Murali, and the question is, how do you set the number of components for the group and the individual subject?
Yeah, that's always a great question. So the easy answer in terms of the group is that we don't have to, because that is kind of done a priori. So in terms of to the template ICA framework, we're completely agnostic to the number of group components. Of course, that is a very important scientific decision. And when I have a number of collaborations where we're implementing this, and that is, step number 1. How do we do that? And we do it the way that anybody else would do it for who's performing group ICA. So we consider the resolution that we want. We, might fit the model a number of times and see how the networks break down. So I am not personally aware of any kind of tried and true method of kind of getting exactly what you want from group ICA. So that's something that is more of a scientific question, and it's not any different from any other like group ICA based analysis. So when you ask the number of subject components, so here we have these additional nuisance components, right? And we actually do have an automated way of choosing that. So you may be familiar with the Minka method. The Minka method is it's kind of a fairly old and established method at this point. The original paper is from like 2008, I think, maybe even going back to like a preprint in 2001. And it is used, I think, in a number of the MATLAB tools for performing ICA. So we use a slightly newer method that kind of built on Minka's method and kind of tried to improve upon it, called PESSL. And so we use an R package where that's been implemented to estimate the number of subject level components. And so we're specifically estimating the number of nuisance components. We also allow the user to specify a maximum number of components. So if you you don't want to have anything more than 200 components, including the nuisance, or if you don't want to estimate any nuisance components, those are things that can be controlled through the functions in the package.
Great, thank you. Also, it's reminding people that if you want to ask a question yourself, you can raise your hand virtually. You can do that by hovering over your name in the participant list and clicking on the hand icon, or down below together with the emojis at the bottom.
Yeah, and Thomas asked about the spelling of the PESEL name, and I can provide that citation as well, because I think that the package should be easy to find, but the reference does not have PESEL in the title, I believe, so I can provide that link.
Thank you.
Hey, Thomas, if I don't remember, please remind me at the end of the talk, but I would be happy to do that.
And yes, we can do that on the Slack channel as well afterwards. I'll try to remember at the end here to move all of the questions and information on this chat to the Slack channel as well. All right. So I'm not seeing more questions. So I guess we can move on.
Okay, will do.
Thank you.
All right, so so now I'm going to jump into a few reliability studies that we've performed. So this basically I'm going to talk about 2 reliability studies. So they both use data from the Human Connectome project, and in both the goal is to assess the reliability of the subject ICs produced from template ICA. The first study compared template ICA just with dual regression, and the second study, which is recent work, compared template ICA with both dual regression and gig ICA. Sorry, I'm going to say gig ICA probably a lot because group information guided is a mouthful. So this first reliability study is contained in our original paper, which I'm again citing here at the bottom. So you can get lots more information from the paper, but I will just show you kind of the highlights here. So here we are using the 25 component group ICA provided through the HCP, and we manually selected 16 ICs that represented known cortical resting state networks that were easily kind of recognizable. And this also, because we're choosing a subset, sorry, gotta stop using my mouse. Because we're choosing a subset of the components, this also kind of tests our ability to deal with the presence of nuisance components, whether they be neuronal or artifactual in origin. So the data that we're using here is ICA fixed data. And so it's been processed through ICA fixed to remove a lot of the nuisance signals. And it's in gray ordinate space, and it's resting state data. So for template estimation, we used 461 subjects from the 500 subject release of the HCP. We concatenated the left, right, and right, left acquisition phase encoding directions within each visit. So for training data, we like to have test retest. So we're going to keep the visit separate, but we're combining the acquisitions within each visit. So we have 30 minutes of data per session for our training. And in our test data set, we chose 20 additional subjects from the HCP 900 subject release. We used the LR run from both visits, and we varied the duration to see the effect of scan duration on our reliability from 400 time points, or about 5 minutes of data, to 1,200 time points, or about 15 minutes of data. So we have a number of ICs. We have 16 specifically, but I'm going to show some, just like probably a single IC for most of the talk. And I actually realized, I think that there's a mistake here, so I apologize for that. So, so here I'm showing the template mean and variance for this particular IC, IC10. And so you can see that the mean, so just don't pay attention to the variance for a minute. I'll explain what's going on there in a second. But looking at the mean, we see that the mean has very similar spatial patterns to the group IC, even though it's based on these noisy estimates across a bunch of subjects and kind of averaging them. It's on a different scale as well, because there's some scaling of the data that is done differently, but that is irrelevant. But we also noticed that we see a few differences between the means and the group ICs in general. So we noticed that the means tend to have more kind of localized features. I'm not sure how clearly it is to see in this particular example, but like you can see here, for example, there's this kind of bright spot in this sulcal fold right here that we don't see in the group IC. There's also the kind of these bright spots right here and here on the medial view, as well as this kind of area of negative engagement here on the medial view. So some of the more subtle features seem to be coming out in these means versus in the group ICs. And this could be because in the group ICA is based on doing a lot of dimension reduction at the subject level before estimating these group average IC maps, whereas the mean is based on using the full amount of data from every subject. So we think that some of these more subtle features are actually not captured in the group ICs and are captured in the template means. So looking at the variance here, I believe I'm showing the wrong component, so I apologize for that. You should see that the areas of engagement, the areas of high variance are very similar to the areas of high engagement on the mean map. So it's the wrong component. I apologize for that. But one thing I wanted to point out here is that you see these kind of missing vertices. These are zeros. So these are areas where our between subject variance estimate is actually negative, and we truncated it at exactly 0. And I'm going to come back to this because this is going to be important for some of our ongoing work. But for now, we're setting those values to 0. Okay, so here's some examples of individual IC estimates that we get from template ICA and then just from applying dual regression. I'm showing 2 subjects. So here, subject 39. I'm sorry, my mouse is so crazy with Google Slides. So I'm showing 2 subjects, subjects 390645 and subject 645450. And the duration is 1,200 time points for each of these subjects. And clearly the template ICA estimates are much less noisy. That's obviously apparent initially, but we also really want to know, that's not the only important question, right? Of course, noise reduction is always good, but we want to know if we're discovering individual features in these template ICA based IC estimates. And I'm pointing out here one area where we do see a difference in the 2 subjects. So here, this area of engagement is a little bit more like inferiorly oriented. In this subject, it's a bit more superior. And we see those same features coming out in the dual regression maps, but they're kind of obscured by noise. And so we see some evidence here that we might be discovering unique features with more accuracy or more clarity with template ICA. But we wanted to go a little bit deeper into that. So we next turn to quantifying the reliability using primarily the interclass correlation coefficient. So interclass correlation coefficient is can be thought of as a measure of individual uniqueness, and it can be increased by either reducing the noise and or preserving the signal. So if we reduce the noise, but we throw out the signal, we're still going to have a very low ICC. So it kind of represents a balance between noise reduction and signal preservation. So especially for a Bayesian method, I think this is a really good metric to use. And so it's simply the ratio of the between subject variance in the estimates to the between and within, or total variance in the estimates. We estimate this separately at every vertex, and it ranges between 0 and 1, with 0 indicating there's no unique subject level information in these estimates, and 1 representing there's subject level information and there's no noise. We can also summarize within an image, so within an IC in our case, using I2C2, and I2C2 is described in this paper that I'm citing here at the bottom. We use a weighted version of I2C2. So I2C2 basically just sums up all these variance components across all the vertices. We introduce weights because we want to emphasize the areas of engagement. So we don't want to overemphasize the noise reduction that we're getting in the background areas. And so we're going to put higher weight on those areas of average engagement. So these weights are determined at the group level using the template mean. So those also range between 0 and 1. So here's an example of ICC maps. And what you can see is, this color scale goes from 0 to 0.5. And you can see that template ICA does have more areas that are kind of this bright turquoise, which represents that 0.5 or above level than dual regression. So just looking at this one IC as kind of an example, we see that not only is template ICA reducing noise, it actually contains more unique subject level information relative to the amount of noise in the data. So this is, I think, a very important point, because we're not only doing noise reduction, we're doing also sufficient signal preservation. And for this IC, these areas where we have high ICC are corresponding to areas of engagement. So this is not just in background areas. So looking at the I2C2, we looked at this across scan durations, and each line here represents one of our 16 ICs, and they're just colored by network. And so if you look, for example, at 5 minutes duration, you see that for every one of these components, the I2C2, or the weighted I2C2, did go up. And that's true for 10 minutes of data as well, and 15 minutes of data as well. But what is perhaps more striking is that if you compare the dual regression reliability with 15 minutes of data, generally template ICA tends to have higher reliability for each individual component with just 5 minutes of data. And so we're extracting a lot more individual information from a lot less data. And I always say, of course, we should get more data if we can, but we should make the most of the data that we have. So whether we have 15 minutes of data or 5 minutes of data, we can improve with using this kind of approach. Okay, so I'll stop there for a brief Q&A, unless, Roger, you think it's better for me to keep going given the time?
No, I think we can go quickly here. There are just a couple of questions I think are related to this topic. If you don't mind, we could
Sounds great.
go over them. The first one is from David Bloom, and he's saying, any concerns with using a data set that only uses subjects 20 to 40 years of age? Also, any reason not to use the HCP test-pretest data set?
Yeah, so I think that it's a very good question. Who you choose to be in your template really depends on what data set you're going to apply this to. So we applied it to the HCP. So our test also comes from the HCP. So we weren't concerned about that. But I'm currently working on some collaborations applying this to people with ALS, to babies, and of course we would not use the standard HCP to come up with those templates. So it is really important to choose a population as well as the data acquisition properties that are representative of your focal population. And regarding the HCP test retest, so there's a balance between the number of subjects you include and the duration of their time series. So with the test retest, we could get double the amount of data for each subject, but we'd have fewer subjects. So I don't have an easy way to say like how many subjects you absolutely need or what duration you absolutely need. But we chose to have a larger number of subjects. But I think that if you had really long scans, like with the retest, and you had a smaller number of subjects, you would probably get very nice templates as well.
Thank you. Another quick comment from Thomas is neat. I didn't know that I2C2 could support SIFT.
I don't know that it does. In fact, so there's an I2C2 package, I think that you're referring to, Thomas, in Neuro, that's available through Neuroconductor. So I think that I2C2, the function is applicable to matrices. So if you can read in the SIFT data using SIFT tools, then you can extract the matrices and just run it through I2C2 that way. And we have I'm going to mention Damon in a minute, but Damon has done, Damon Pham has done pretty much all of the software development for the Sifty Tools package and has implemented an impressive number of S3 classes. So you can run like as.matrix on a Sifty file after reading it in, just get the data out, even though it has all this additional information. So it's pretty convenient to grab the data, like kind of the matrix of data from a Sifty file.
Thank you. So here there is a question that's from Armin about what was the within and between values in the ICC. What were the within and between values?
Within and between. So what were they? That's a good question. I don't have those results. But if that's something that you'd like to know, I can track them down for our current reliability study. I'm not sure how easy they'd be to track down here. But it is an interesting question because the within tells you how much noise there is and the between tells you how much signal there is. So we really like to think about the ratio of those things, but it's also pretty interesting to look at, what is actually going on under the hood. So I suspect that the template ICA had much lower, sorry, I keep accidentally flipping around, had much lower within subject variance and maybe even slightly smaller with between subject variance if I had to guess. But that on balance, the ICC was higher.
Thanks. And one last question from Armin is if you compared with Gig ICA results or you're planning to?
Yeah, that's the next reliability study.
Great. Okay, so let's move on to that then.
Okay. So this is really our current work. So Damon Pham, who's an absolutely fantastic post-bac researcher who, if he applies to your graduate program, you should absolutely consider him strongly because he's done amazing work with my group. So he's been working on this reliability study comparing template ICA to both dual regression and group information guided ICA. And I'm going to show those results, and I'm also going to show a methodological improvement that Damon developed as part of this analysis. So here we're using the 100 component group ICA from the HCP, and those have been grouped into 7 YO networks. Reference for those networks are here at the bottom. And here we're actually using the minimally preprocessed data, again in a gray ordinate space, again resting state. Our training data set includes 154 subjects from the HCP 1200 subject release. We're using just the LR runs for both visits, and so we have about 15 minutes of data per visit. And then our test data set includes 104 subjects, also from the HCP 1200 data set, separate from the training subjects, but they're sex and age balanced with the training set. And then we're using the LR run for both of those visits as well, and again, 15 minutes of data per visit. Okay, so here, again, I'm showing just an example of one IC, the group IC, the mean across subjects, and the template variance, or the between subject variance. So again, the mean IC looks a lot like the group IC, but with more of these kind of localized features. So you can see here, we're getting this area of kind of negative engagement that we really weren't seeing in the group IC, and then similarly on the lateral view, we get more of these kind of specific features popping out. The between subject variance, first of all, again, we see that the areas of high average engagement are the areas where we're seeing a high amount of variance between subjects. And in these background areas, we're seeing either low estimates of the between subject variance or actually negative estimates, which we truncate at 0. And again, I'm going to come back to that. But we see a lot more of those negatives appearing in these estimates, or these truncated zeros. And we believe that's because we're using less data this time. So we're not using 400 subjects, we're using approximately 100 subjects. And we're not using two runs per session, we're using 1 run per session. So there's a lot more noise affecting these estimates. So here's an example of the IC estimates that we get from 2 subjects. when we apply template ICA, dual regression, and group information guided ICA. So with template ICA, you see there's like a large amount of kind of shrinkage towards 0, which is the average, close to the average value in these background areas. And the area of activation is kind of seen in this bright yellow. And one feature that I want to draw your attention to is that in this particular subject, we have this kind of long tail, for lack of a more scientific word, in this motor region. And in this subject, we don't have that same intense long tail. And you see that same feature coming out in the dual regression maps and in the GIG ICA maps as well. The dual regression maps and the group information guided ICA maps tend to be somewhat roughly, have roughly the same amount of noise we've seen. So we'll get into more quantitative comparisons of their reliability, but they're both kind of contain much higher levels of noise than template ICA. And then looking at 2 visits from the same subjects, for the same 2 subjects, we see that that tail feature that I mentioned earlier is really preserved across the visits. So this seems to be a real feature of the subject's functional topology, as opposed to this other subject where we see that we don't have that intense tail. So this is a feature that we're kind of see how well we can estimate with these different methods. So to quantify reliability here, we're still going to look at ICC, but we're also going to look at test-retest mean squared error. We're using a normalized version to remove any kind of difference in the scale or the units across the methods. And the reason that we're looking at mean squared error as well as ICC is because ICC tells you about uniqueness, but uniqueness is not the only kind of important feature of these IC maps. Because they serve as the basis for estimating functional connectivity in many cases, we also want them to be accurate. So for example, in dual regression, one kind of, I think there's a bit of an elephant in the room with dual regression, which is that when you estimate the temporal mixing matrix, you're estimating it based on the group ICs. So that first regression step relates the group ICs to the subject level data, and that's how you get your temporal mixing matrix. So it's kind of equivalent to like a weighted combination of the vertex or voxel time courses weighted by the group IC intensities. And then the second step, you get the subject level IC maps, but those are not forming the basis of estimating functional connectivity. And that's why dual regression actually produces pretty reliable estimates of functional connectivity, because you're using these really highly reliable group IC maps. So we do care about accuracy as well as uniqueness. And so mean squared error can tell us about accuracy, whereas ICC tells us more about the uniqueness relative to the amount of noise. Okay. So looking at mean squared error first, looking at just this one component, the motor component that we've been looking at, you can see that template ICA does have lower mean squared error relative to dual regression and GIG ICA. The differences are a bit subtle on this color scale, and we're using just within the mask because of the normalization. So we're normalizing by the kind of average intensity of the engagement within this mask. And so if we average within that mask, so we're looking at just at these areas of high engagement, for each network, this is the average normalized root mean squared error across all vertices within the mask for that network. Every network has like a random color, but they're grouped by the 7 Yo components. So you see template ICA has pretty low mean squared error. Dual regression tends to be much higher, so this is very consistent with what we've seen earlier. And then GIG ICA tends to be somewhere in the middle for most of these networks. So it's much closer to template ICA and much than it is to dual regression for the most part. It tends to be a little higher than template ICA. And so that tells us that even though it looks noisier than dual regression, in general, it's doing a better job of estimating the signal consistently than dual regression is. One interesting thing as well is in this cortical, template ICA has much lower mean squared error than either dual regression or group information guided ICA. And for some reason, group information guided ICA actually has a very large mean squared error in subcortical areas. Okay, and then we started to look at ICC, and this is where things got really interesting. So we look at template ICA, ICC maps. So red is low, yellow is kind of in the middle, around 0.5, and then blue is like perfect, it's 1. We see that overall, template ICA has higher ICC kind of overall, but in the areas of engagement, we were actually seeing that it seemed to have maybe lower ICC than dual regression or gig ICA. So the uniqueness was being to some degree reduced with template ICA. So we started to wonder if we were over shrinking, if we were overly constraining the model to shrink towards the group, which can cause that loss of individuality. So we considered an alternative method of estimating our template variance. So I said I would come back to this because we saw all those kind of negative values that we truncated at 0. So our template, our variance estimation or template estimation in general is based on a very simple measurement error model. We assume that repeated estimates for each subject can be modeled as having a visit effect, a subject effect, and a residual. And the residual is assumed to be independent and independent from these other terms. So the between subject variance of this kind of truth for each subject Z, that's what formed our template variance. So we want to estimate variance of Z. And so there's a couple options that we have. The first is an unbiased estimation approach. But if we do unbiased estimation and the true values are close to 0 or there's a lot of noise in them, we can get negative values. And that's what we were seeing in those maps. And so whenever we get negative values, we know negative variances don't exist, so we would truncate them at 0. And that was what formed the original approach and everything I've shown you thus far. Another option, though, is to use a biased ANOVA-based estimation approach, where all of our values would be greater than 0, and they would converge to the true variance, converge kind of down to the true variance as we get more and more data, but they will be a little bit upwardly biased. And this is something that was attractive to us because that way we would be avoiding underestimation of the variance. Right, so anytime we're estimating things, we're going to do a little overestimation, a little underestimation. This way, we're kind of building in a buffer so that we're going to, in general, overestimate the variance, not underestimate it. And that's going to lead to less shrinkage towards the group. So it's going to preserve more of the individual features. Okay, so we tried this new approach, and I'm going to show you its effects. So first of all, if we do the unbiased variance estimation, this is what our template variance looks like before we set all these missing values or these negative values to 0. If we do the ANOVA-based, we see that overall we get kind of higher values, but we get no zeros, nothing even really close to 0. This is a scatter plot comparing the unbiased standard deviation to the ANOVA standard deviation, and you can see that in general, I mean, everything is above this equality line. And you can see also that there's this huge kind of spike at 0 for the unbiased because those have all been truncated, and now those are taking on a range of values. So what is the effect on the IC estimates? So on the left, I have the visit 1, visit 2 for both subjects using the unbiased template variance and template ICA, and on the right, I have our updated approach. So you can see there's a little more noise coming out, and that's kind of a consequence of having this lower template variance because we're doing less shrinkage and doing less noise reduction. But you actually see that this feature, this kind of long tail, is maybe being, it's more intense if we use the ANOVA-based variance estimation. So it's maybe being more true to the subject individual features than our original approach. And here again, on this subject, you still see that long tail does not come out. And in fact, the tail might be a bit shorter than in this unbiased approach. So in general, we're seeing a little more noise, but a little bit more uniqueness. Okay, so looking at the ICC, here I'm showing just first this motor component we've been looking at. It's a little hard to see the differences, but we are seeing, if you kind of compare them on top of each other, you can really see that we are seeing higher ICC with this ANOVA-based variance template variance estimation. So you see a little bit more like yellow versus orange. You see a little bit more blue popping out as well. It's a little bit more apparent in some of the other components. So I'm also showing a default mode component here as well. So here you can see that there's just a lot of areas that have gone from kind of almost orange and red to, so those are like values, say close to 0.2, 0.3 ICC, to more yellows, so close to 0.5 ICC. So we do seem to be getting more uniqueness relative to the amount of noise reduction using this updated approach. And then looking at the mean squared error, this is where we pay a bit of a cost. So the mean squared error is more sensitive to noise. And so because we have more noise in our ANOVA-based approach, you see generally these mean squared errors go up a little bit, but not by a ton. The biggest increase we're seeing here in the subcortical areas. And so I wanted to show just an example of that. So this is two subcortical components. We have a IC79 is in the putamen primarily, IC84 is in the thalamus. So here I'm showing the dual regression mean squared error, the gig ICA mean squared error, where brighter colors are higher or worse mean squared error. And then the template ICA using the unbiased versus the new ANOVA-based approach. And again, you see this, that there's clearly an increase in the mean squared error with this new approach, but it's still less than what we see with dual regression or group information guided ICA. So template ICA is still doing the best job in the subcortical areas. So we believe that this price is worth paying for the additional uniqueness that we can get, and just avoiding over shrinking towards the group, because that's kind of the biggest concern that I hear when we talk about Bayesian approaches, is how do you know you're not just throwing out all the subject level information? So here we're being a bit more conservative now. And then finally, I think this may be my last slide in this part of the talk before just a quick recap. I wanted to illustrate how we can do inference. As I mentioned inference previously, but we haven't really shown any examples of doing it. So inference, again, could serve at least two purposes. The first is that you might want to quantify the size of the engagement. So we might want to say, like, some subjects have really large engagements, and particular networks, some subjects have smaller engagements, and that might relate to some kind of disease status or treatment effect or development, any number of things that we might want to relate these maps to. And so by doing inference, we can actually quantify the size of the engagement. We can also, again, use this to perform principled thresholding. So thresholding IC maps is typically a bit ad hoc. So here we could, for example, identify the area of engagement for every subject, and then maybe look at the union across all subjects to come up with a principled mask that would include all of the engaged areas for any subject. So from template ICA, we can get the estimate of the IC for a particular subject, as well as its standard error. And then we can simply perform a t-test at every vertex, and then follow that with multiple comparisons correction to identify the areas of significant engagement. Here we're using FDR control with a Benjamin E. Hochberg correction, or Benjamin E. Hochberg procedure, with the Q set to 0.05. And so what you see here is that for this subject, on visit 1 and visit 2, we are, again, apologies for that, we are detecting that longer tail of engagement in both visits. Whereas for this other subject, we don't see a lot going on in this area. So we see a much smaller area of engagement. And that's just quite consistent across visits. And so it seems to be that we are able to kind of uncover these unique subject level features, at least with some degree of accuracy. Okay, so I planned a Q&A here, and I think I literally have one slide after this. So just to give you an idea of where we're at.
Sounds great. Yeah. All right, so I have a few more questions coming in. I think There was a question hanging in from the previous part that I missed. It's from Armin asking if you had looked at brain dynamics.
We have not looked at brain dynamics, but so with template ICA, I should have mentioned this, but we do get an estimate of the temporal dynamics, and so we could certainly do dynamic functional connectivity with those temporal activation patterns. I'll mention this in a minute, but some of our ongoing work is looking at putting empirical priors on that functional connectivity as well, so that we can do kind of our best job at estimating the functional connectivity.
Right, got it.
So this is not at all incompatible with the dynamic connectivity approach.
Mm-hmm.
But we do assume that the subject's actual functional areas do not change over time, at least not within a session.
Right. So another question is on regarding slide 24. This is in the second part of this last part of the talk. And it's asking if the three plots are normalized similarly.
Yes, so this was actually a big challenge. So template ICA and dual regression are actually on the same scale, and I apologize, I should have the scales here. There's just an oversight. So template ICA and dual regression are both designed to be comparable to each other as well as to the group IC maps. But gig ICA has a different approach. So gig ICA, first they scale the data a bit differently, and we actually tried implementing our data scaling approach, and we actually think it improves things a bit for gig ICA. But that being the case, even when we scale the data the way that we do it for template ICA and dual regression, and even when we also scaled the group ICs in the same way, the results were not on the same scale. And it turns out that we think gig ICA is enforcing that every map at the very, I think maybe the very end, and somebody here may know more about it than I do, or maybe more familiar with it, turns out that the spatial maps all have standard deviation of 1. So there's no units, that you can preserve if you at the end fix the scale. So the gig scale is not comparable to the dual regression and template ICA scale. So we use the quantiles to try to find a reasonable way to at least visualize the maps on a comparable scale. It was a real challenge when we were looking at mean squared error, and that's why we developed this normalization procedure so that it's not sensitive to the scale. So we assume that within a method, all the subjects are kind of on the same scale, but we assume that there can be differences across methods in terms of the scale.
I see. Thank you. There is another question from Thomas. This is on slide 32. And he was asking if there is something you could do to cross-check with LGI.
I actually don't know what LGI is. Can you explain that?
Let me see, would Thomas like to come up and ask the question himself? Maybe. Can ask you to unmute. We do not hear you, Thomas.
It's a local gyrification index. So it's a measure of how deep inside of a gyrus a particular location is. Okay. I would be really curious to hear your thoughts on how that could relate. If gyrification relates to the quality of alignment across subjects, then that could be something that we're influenced by, because we assume that the subjects are aligned properly, and of course they never align perfectly, but that is an assumption that we have to make, just as almost any other vertex or voxel based analysis of fMRI data does. So yeah, I'd be curious to chat more about
that. Thank you. And then the next one is from Armin. about slide number 35. Have you calculated similarity with the reference map for all approaches?
No, I haven't. So for certainly it's going to be higher for template ICA than for dual regression, but it's not clear how we could do that for group information guided ICA using the data scaling that's done at the end. And so just like to talk a minute more about that. When you fix the standard deviation at 1, it actually, so even if your group IC maps have standard deviation 1, that makes it so that you can't really compare with those group IC maps anymore, because the more noise you have, the higher your standard deviation is. So we can think of the distribution of intensities as kind of a mixture distribution of the signal and the noise. And the noise tends to be really high variance, and the signal tends to be much lower variance. So if you have kind of a mixture of two normals, where normals or whatever else it may be, where you have a low variance like signal component and a high variance noise component, if you fix the signal, if you have the group IC maps and you assume there's only signal, and you fix that standard deviation at 1, and you fix the mixture distribution at standard deviation of 1, your signal component of that mixture distribution is actually will have a much lower standard deviation, right? Because the noise, the presence of the noise is increasing the standard deviation. Relative to just the signal distribution. So it makes it so that it's really difficult to compare with the group information, the group ICs.
So a quick follow-up to that was, could you use correlation instead to compare?
Correlation, yes. So correlation, gotcha. Correlation with the group ICs. Yes, we could look at correlation. I mean, definitely, I think we're going to see template ICA is the highest, but again, like, we want to think about uniqueness, not an accuracy to the subject level functional areas. So this is something that I try to be really sensitive to, because I often take Bayesian approaches, which do shrinkage towards the group, that I don't want to reward my method for just replacing the subject level estimates with the group average. So I usually try to think about what metrics kind of respect also the subject uniqueness. So I think we would see that we have really high similarity because we have a much higher amount of noise reduction, but I don't think that's going to tell the full picture. I will say that in our simulation studies, we did use correlation with true subject level ICs as a measure of accuracy.
Yeah, so Armin is agreeing to your point here that the idea is to have higher within subject similarity, but less similarity with reference.
Yeah. So I guess the issue is that when we look at similarity with reference, it's a combination of the signal similarity and the noise, the amount of noise. So even if your signal is identical, if you have a bunch of noise, you'll have a low correlation. And if your signal is different because you're actually estimating the true subject level signal, but you have a bunch of noise, again, your correlation is going to be low. So it's kind of hard to tease out what is causing the correlation to be high or low. And that could be either a good thing or a bad thing, depending on like how much noise there are in the estimates.
I think those are very interesting insights. Thank you. Yeah, so we're done with questions. And we can move on to the last part. Thank you.
Great. Thanks, guys, for the questions. Appreciate that. So I think just 2 slides to wrap up. So one of the nice things about this template ICA framework is that it's easily extendable to incorporate additional sources of information. So the first extension that we worked on, and we actually, I don't have the citation here, but we have a preprint out on this first extension, is to incorporate spatial priors to specifically on the cortical surface, to encourage or to account for and leverage the dependence along the cortical surface. And so specifically, we're assuming the prior on the deviation. So those deltas in our original model, those were sort of implicitly assumed to be independent spatially. And so that is not true. They're smooth. And so we built this spatial model that also imposes a spatial prior on those deviations. And with the spatial prior, I did not include the slides in the main talk, and I think that was a wise decision looking at the time, but I have a lot of results on that in the appendix if anyone's interested. But generally, we see that we have an increase in statistical efficiency to estimate the subject effects, so that's accuracy. And then we also have increased power to detect true engagements in individual ICs, as well as the deviations between subjects and the group average. It is quite computationally intensive relative to template ICA. So it's something that I would recommend using if first you're interested in cortical surface analysis specifically, like not subcortical and not volumetric. But also if you have a short amount of data for every subject, then the benefits could make up for the computational challenge. And when I say it's difficult computationally, I mean it's not days, it's hours. So it is doable, but if you have a large group of subjects, it could take a little bit. Anyway, it is implemented in the template ICAR package, and I'm happy to talk more about that if anybody's interested. And then we have two ongoing projects. The first is what we call functional connectivity template ICA. So in addition to having an empirical population prior on the spatial maps, we also are incorporating an empirical population prior on the functional connectivity, which is basically, it boils down to assuming a prior on the covariance of the rows of the A matrix. So each row of the A matrix is a vector of length Q. And so we can think about it as following a multivariate normal distribution with the covariance encoding the functional connectivity between the components. And by Q, I mean the number of template components. So we can estimate that, and the hope there is to also improve the estimation of the functional connectivity between the components. And then the final extension is what we call diagnostic template ICA. And so the idea here would be that we have multiple groups. So we have a disease group and a healthy group, and we estimate separate templates, both matched to the same set of group ICs, but separate means, separate variances. And by fitting the model on an individual subject, we can, in theory, learn each subject's true group membership based on how compatible that subject is with that particular population. So again, ongoing work. So we'll hopefully be getting a number of additional papers out in the coming months and beyond. And I haven't even mentioned our applications, but this is being used in a number of really fascinating applications, ranging from the effect of psilocybin on the organization of the thalamus, to the organization of babies' brains, to the effects of ALS. So just to recap, template ICA is an empirical Bayesian hierarchical ICA framework that has a number of features, including noise reduction, matching subject ICs across subjects and studies, producing more reliable subject-level IC estimates. Inference can be performed to quantify the size of engagements and to threshold IC maps. And finally, the model can be extended to incorporate additional sources of prior information. And our new ANOVA-based template variance estimation approach avoids the possibility of overshrinking towards the group average. So with that, I'd like to just say thank you so much for your great questions and your attention, and for, again, to Rogers for the invitation, and to highlight both my all my collaborators as well as my funding agency, NIBIB.
Nice, thank you so much. All right, let's see if we have one more question. And given the time, I think we're probably going to go and make that the last one. So this is from Armin again, and he's asking for reference-based functional connectivity. Are you using static FNC, or is it dynamic?
Yeah, we are using static functional connectivity for now. It would be interesting to extend it to dynamic, so that's another extension that we could definitely think about, and then potentially even like identify those states within the model. That'd be really neat. nice to have a computationally efficient framework so that we could kind of stretch it in different directions that might be more computationally challenging. So in terms of how we estimate it, so you're saying we have one sample per subject. In our training set, we always separate it into two. We could do more, but we kind of typically have two. So we either have true test retest data, or we take our data set and we split it down the middle in terms of duration, and we separate it into pseudo test retest. So that's really essential for the template estimation for both the spatial components as well as for the functional connectivity. So in terms of how we estimate it, it actually is a bit tricky. This is more challenging than for the spatial maps. So the prior on the covariance is an inverse Wishart prior, and that prior has two parameters, one that's related to the average and one that's related more to the variance. And that variance, though, is common across all independent components, or all pairs of connectivity. And so estimating the component that has to do with the average was easy, because then we can just average across all of our estimates, and that gives us an estimate of the average functional connectivity. But estimating that variance was something that we thought about, the parameter that controls the variance, something we thought about a little bit more, because it turns out that different connections have different amounts of variance across subjects. And here I'm talking about the between subject variance. So after we account for noise or within subject variability, how much is explained by the between subject variability. And we chose to set that parameter such that it would kind of allow for more variance than what we observe empirically. And this is going back to the same kind of concept we were talking about earlier with the ANOVA biased variance estimation approach, that we don't want to overly constrain the model to be shrinking too much towards the group. So we chose, we kind of estimate that parameter separately for all connectivity pairs, and then we choose some upper quantile across all the connectivity pairs.
Right. And this is, you're describing the estimation of the priors, right?
On the prior, and this specifically for the functional connectivity template ICA. So we have the multivariate normal prior on each row of A, but then the tricky one is having the hyper prior on the covariance of that multivariate normal. Mm-hmm. And how do you estimate the parameters of that using the training data that you have?
And so just to make sure I understand, the covariance is dimension of component by component?
That's right, Q by Q, yes. Yeah. So we estimate the covariance, so our priors on the covariance, but then you can just do a simple transformation to get the correlation.
Mm-hmm. And then within the single subject, once you have those priors, each type point acts as a sample, right?
Right.
In estimating the covariance,
exactly.
Or the FC.
Mm-hmm. Yep, exactly.
Thank you. Yeah, I think that addresses the other question that Armin had here. Yeah, this is very, very fascinating, very interesting. That one is already published or coming out?
No, this is ongoing work.
We're
actually still in fairly early stages. We're developing the computational estimation strategy. Mm-hmm. So we think it's gonna, we were working on an EM algorithm building on our previous work, but that inverse Wishart prior makes things all much more complicated. And so we're looking at a Gibbs sampler now, which basically allow you to update the A and its kind of parameters in one step, and then update S and its parameters in another step, and kind of alternate between those two. So so that's very much ongoing work, but we're working hard and quickly on it. So we do hope to have it out in the next several months.
Awesome. That's very, very encouraging. Thank you. All right, so that was the last question for today. We'll be wrapping up now. I just wanted to thank Dr. Mejia very much for this great talk, and thank everyone else for attending today. We will continue this discussion here, maybe for another minute after stopping the recording. And everything else later on is going to be via Slack. So thanks to everyone, and I'll stop recording now.
Thank you so much for all your great questions.
Sure. Thanks.